Buenas tardes a todos. Un placer poder contar con su presencia a nuestro segundo webinar correspondiente al uso responsable de los antimicrobianos, moción organizada por la Organización Mundial de la Salud y fortalecida por diferentes actores público-privados, como lo son en esta ocasión la Asociación Latinoamericana de Avicultura y la International Poultry Council. Recordarles que pueden utilizar la traducción simultánea y escucharlo en el idioma de su preferencia. Asimismo, se les indica que pueden realizar sus preguntas por el chat o por la opción de preguntas y respuestas. Damos inicio a este webinar con las palabras del señor Ricardo Santín, presidente de la International Poultry Council.
Hola, Dania, muchas gracias. Hello to everyone. I would like to greet my dear friend and my boss, President Maria de Rosario de Penedo Falla, President of ALA, that is a co-producer of this event. I'd like to greet Dennis, my dear friend Dennis, that is the responsible for Transform in IPC. Nicolo Cinotti, my Secretary General, and Marcelo Oliveira, and Dania, of course, our General Director from ALA. It's a pleasure to be here again for the second edition with Dr. Tom Schock to reinforce the commitment that we have made as IPC, as ALA, and also as associations, country associations in the world. And here we are making this with ALA, or to bring this theme on discussion and reinforce our commitment on working on the responsible use of the antimicrobials. And then I would like also apologize that I'm traveling and here in a hotel, living at this moment for a travel, but I'd like to wish a very good event. And I'm sure that these things like this, the works like this, will spread in the world a responsible use of antimicrobial, and we will harvest these fruits of this work. Thank you so much. Have a good event for everyone.
Muchísimas gracias, don Ricardo, por sus palabras. Quisiera darle la bienvenida a nuestros conferencistas de hoy, el señor Nicolo Cinotti, secretario general del Consejo Internacional de Avicultura, al doctor Thomas Schreier, microbiólogo experto en antimicrobianos y responsable del proyecto TRANSFORM, y el señor Dennis Erpolding, asesor científico y comercial para la International Poultry Council y el proyecto TRANSFORM. Asimismo, agradecer a Cobb Vantres por su apoyo en este webinar con la traducción simultánea y a Marcelo Oliveira, coordinador del Instituto Latinoamericano del Pollo, de ALA. Para dar apertura a este webinar, le cedemos la palabra a la presidente de ALA, la señora María del Rosario Penedo de Falla.
Buenas tardes a todos. Gracias, Dania. Y gracias a todos los que coordinan este grandioso evento tan útil para la avicultura latinoamericana. Nuevamente es un honor darles la bienvenida a esta importante serie de webinars organizada por ALA e IPC. Hoy, más que nunca, es nuestra responsabilidad y es muy clara. Unirnos como región para implementar prácticas que reduzcan la necesidad de antimicrobianos en la producción avícola, protegiendo así la salud animal, humana y ambiental. Las herramientas están aquí. Las guías y enfoques, como el proyecto TRANSFORM, nos marcan el camino. Pongamos en práctica estas iniciativas trabajando juntos hacia un futuro más sostenible y seguro para todos. Manos a la obra. Gracias.
Muchísimas gracias, presidente. Para dar inicio formal a este webinar, le cederemos la palabra al señor Nicolo Cinotti, secretario general del Consejo Internacional de Avicultura. Nicolo, go ahead.
Thank you, Dania. Good evening, good morning, good afternoon, everybody. Let me first thank my dear President Ricardo Santina and President Maria de Rosario for the words of introduction to this event. And next slide, please. Thank you to all that are joining us again today for the second webinar on the antimicrobial resistance and the Transform project. Last time we approached the antimicrobial resistance from a global point of view, giving you an understanding of what is going on around there, how we got to this point in the discussion, and what is the role of IPC. We touched on the role of the international organizations such as the World Organization for Animal Health, or WOA, the Food and Agriculture Organization, the Codex Alimentarius, and the quadripartite AMR multi-stakeholder platform. Those are stakeholders, international stakeholders, where IPC is having a role and is playing his role at global level. And everything that IPC is doing it thanks to the leadership that its members have shown in the past years. Next slide, please. I would like to remind you again that IPC is the global association representing the poultry meat sector, so broiler, turkey, ducks, and other species. And ALA is a beloved member of IPC, and we are proud to represent ALA as well before those international organizations. And we are recognized as the voice of the poultry meat industry globally. IPC has always acted basing on its member priorities, and I already mentioned last week that in 2016, AMR clearly raised as a priority for our sector. And IPC took the lead in 2017 and 2019, releasing its antimicrobial stewardship and best practice to reduce the use of antimicrobials in poultry production, which led IPC to have a key role in the Transform project. So we recognize that action starts at farm level. So that's why we have organized these webinars to show you what we have discussed yesterday, how that is impacting what you are doing on a daily basis in your farms, in your daily activities. Last week, we forgot to mention that it was Antimicrobial Awareness Week, the World Antimicrobial Awareness Week, and it was really timely that we launched this series of webinars during that celebration. And it's really important. Probably those of you who are active on LinkedIn have seen a lot of posts and discussion around antimicrobials. This is a subject that is here to stay, and our industry will be asked to give answer to certain questions. What we are doing with Transform project, and I would like to congratulate ALA and ABPA, Ricardo is here, for their leadership in endorsing the IPC antimicrobial stewardship, because what we're doing with Transform is basically staying ahead of issues. We are trying to decide our own destiny before others will put restriction, measures, regulation that we might not be ready to comply with. So I would like to congratulate with the private sector leaders, the already mentioned ALA and ABPA. Next slide, please. I thought there was the slide with all the endorsers, but anyway, here you can see the IPC antimicrobial use stewardship and the best practice to reduce the need for antimicrobials in poultry production. The first one released in 2017 and the second one released in 2019. And as you can see, the second one, best practice, we have issued those guidelines, those best practices together with the OIE, as it was known at the time. Those two documents are really agile and easy to read and understand documents that you can find in IPC website in the resource section. Otherwise, you can easily ask ALA or IPC directly, and we will be more than happy to provide you with a copy of those two. Next slide, please. As I was saying, I would like to congratulate all the 26 organizations that are supporting the Transform initiative, representing over the 40% of the global poultry meat production. This is something that we started 3 years ago, but I can tell you this is having a real impact. All those entities listed there can tell you how glad they were they endorsed the Transform initiative, because what came after? Transform was selected by Fortune as 2024 Change in the World list, which is a big deal. Every single time that someone is asking you, okay, what you're doing when it comes to AMR, they can prove, beside what they're doing at national level, that they are also comparing and acting at global level. So giving an understanding that the poultry industry is not only looking at AMR national level, but we have a broader view and a broader approach to the topic. I would like to stop here. I want to leave the floor to Tom Shyrock, he's a science advisor for the Transform project, and he will now dwell today a little more in depth into the technical aspect of the antimicrobial resistance. So thank you so much. And Tom, the floor is yours.
All right. Thank you very much, Nicolo. And it's my pleasure to be here this afternoon to share a little bit about antimicrobial resistance, the science, the risks, and the impact. So I'm going to go through these slides fairly quickly because I have a lot of them. But fortunately, there's a Q&A session. So if you have further questions, please write them down so you can ask them at the end. So let's start with our topics that we're going to cover here in this section. First is antimicrobial resistance, the issue and the basics. So I'll go over this. It's going to be a little bit sciencey, if you will. I'm a microbiologist, so I love my bugs and I love my drugs. So you'll hear a lot about antibiotics and bacteria. And then we'll see how regulation, government regulation, and risk analysis are used to specifically guide practitioners, the veterinarians, and farmers in the way that they use antibiotics that will result in interventions that are appropriate for poultry diseases. Next slide. So here's the key definitions. They're very lengthy, but they're in English, so you can have them translated later if necessary. Basically, when we talk about an antimicrobial, it's a very broad term, and it can include any sort of product that targets bacteria, viruses, fungi, or parasites. So it's a wide-ranging term that we use very frequently. Sometimes it's used as though it were the word antibiotic, which is the next definition on the screen. And this is a very specific type of antibiotic product where it's targeting bacteria. And mostly these are bacteria that cause disease in humans or animals. And then lastly, anticoccidials are listed. And although they're not the focus of the talk today, because ionophores are probably most well recognized in the poultry industry, and they do have antibacterial effects, we put them up here on the screen for to be all-inclusive. Next slide. So antimicrobial resistance, the issue. This is a well-traveled wiring diagram that I've used for many, many decades to describe AMR, antimicrobial resistance. And while it's very complicated, what you see as you look at it, the boxes and the circles, there's arrows, there's lines, there's colors. We're going to focus in on the lower right side where it has humans. Humans are the ultimate focus of what we're trying to do to protect public health. Now, as we backtrack just to the left, the box to the left is yellow and the box to the left of that is green. We have poultry in red. Poultry is the focus of what we all do with IPC, with ALA, and other organizations. So that's the thrust of the discussion and relevance for all the talk today. So basically, what we're going to talk about is the transfer of antimicrobial resistant foodborne pathogens from the poultry through the processing, meat production, and being ingested by humans. So that's our 3 steps that we're going to emphasize quite a bit this afternoon. Next slide. So this all falls within the realm of One Health. One Health has become recognized. We've mentioned the quadripartite, and even on that epidemiology slide, humans, animals, crops, and environment. So we're going to really focus in here on those 3 boxes that I just described by the colors. But we're also going to note that the One Health focus is really on the subset of foodborne antimicrobial pathogens, whereas the veterinary and the farmer tend to focus on the bacterial diseases in the birds and the flocks, which is only natural. We'll talk a little bit more about that as we move on. Next slide. Oftentimes, I have heard folks get residue and resistance confused. So I thought it worth one slide to at least have as a placeholder to separate those two. Residues are basically a small amount of antimicrobial product or chemical that remains in an edible tissue after the main part has been used, gone away, or degraded. And these are regulated by established safe tolerance standards. And finally, those would be, as indicated in the top black box, by an average daily intake or by an MRL, a minimum residue limit, so that you can be assured of safety when those are consumed, should they be used on farm. Resistance, in red here in the second bullet point under definitions, it's a natural biological process of microorganism survival, which enables the bug to survive the attack by the drug. And I'll give you more details on that as we go on, and then conclude in that bottom red box about resistance. The way that this is controlled is through government regulations around the use of the products. And that is where TRANSFORM relies on the government regulatory authorities in product approval to provide the user instructions for those valuable products. Next slide. So we've kind of come back again. You'll see this slide time and time again. The goal here of TRANSFORM in the lower left-hand corner is to reduce foodborne resistant bacteria, ones that can actually pass from poultry, because that's what we're focusing on today, into the meat, which could be then ingested by humans. So the goal by reducing that first level input should result in a decrease in contaminated meat products, which should result in a decreased amount of human disease. So that's the high level thought process going on here about the risks. Next slide. Now it's my fun part to talk a little bit about the bacteria, or the bugs, if you will. I'll keep this fairly short, but I want to set up for you just some of the pieces of what a bacterium has that makes it click, that makes it work. And then we'll talk about the antibiotics and how they impact all the behavior and the metabolism and the growth of the bacteria. So starting from the outside, we have the red flagellum. That's like it enables movement of the bacteria. The little wavy blue lines, those are called pili. They allow attachment to intestinal cells so that the bugs don't get moved out of the GI tract. They adhere to the cells. And then starting with the capsule, the cell wall and cell membrane, those comprise the external environment. They're the shell, the casing for the bacterium. It's a single cell after all, so it's all self-contained. Within that casing then, you have the cytoplasm. It's a watery matrix, and we have one chromosome, and we have extra chromosomal DNA called plasmids. Plasmids love to acquire antibiotic resistance genes. They're very good at that. So more on that later. And finally, the little dots are listed right there. Then those would be the ribosomes, as well as the protein synthesis machinery for the cell. So those are some of the key things that just have a mental image of what a cell is looking like. Next slide. So here we have a cross section of a generic bacteria, for example. On the left, at the very bottom, you'll see it says gram-positive. That's one type of bacteria, distinguished by that cell membrane and the cell wall. Gram-positive have a very thick cell wall. On the right side, gram-negative, and this is a very complicated membrane. So that's important in terms of getting antibiotics to their targets or preventing them from getting to their targets. So as you can see, there are 4 boxes of text here, 4 key targets for antibiotics. We'll start here on the very left side, where its synthesis of cell wall can be inhibited by antibiotics, such as the penicillins and so forth. As we then move up toward about the 1 o'clock position, there is the inhibition of protein synthesis. Proteins would be things that are made from amino acids, such as enzymes, structural proteins, etc., key to survival and pathogenesis of the bacterium. And then the next box down there, inhibition of DNA or RNA, messing up the ability of the cell to reproduce. And finally, folate synthesis can be inhibited by the sulfa drugs, and that is more of a metabolism interference. So those are the key targets for the antibiotics. So what can the cell do about that? The next slide tells us that there's the spectrum of antibacterial activity. So for the gram-positives, they are only affected by some antibiotics, such as penicillin, but not from some antibiotics that only hit the gram-negatives. So there's a natural or intrinsic resistance going on here. The gram-negatives, for example, E. coli, they're sensitive to colistin, but they won't be active against by penicillin. So those are called narrow spectrum. So they're only targeting that gram-positive or the gram-negative. The bottom, you see broad spectrum, like tetracyclines, they don't care. They hit both gram-positive and gram-negative. So in terms of a prudent use or appropriate choice of an antibiotic, you want to minimize exposure to an antibiotic that you would not basically be affecting. So there's reasons to use a narrow spectrum when possible. Next slide. So here's the resistance mechanisms. Again, it's the same cross-section, the same gram-positive, gram-negative on the left and the right side, respectively. Efflux pumps. These are basically pumping. It's like trying to bail the water out of a boat. They're trying to get rid of everything that comes in that's toxic, that's not needed. So pumping the antibiotics out is one defense mechanism. On the right side, binding site modification. It's like changing the lock and key from what was there before. So this antibiotic will not bind to the site on the ribosome, and thus there's no inhibition of protein synthesis. And then finally, at the bottom, the DNA can also be inhibited as well. And that can be avoided by, again, enzymes that actually degrade or inactivate the antibiotic. So there's a war going on, antibiotic versus bacterium. Next slide. As I mentioned earlier, bacteria love to acquire resistance genes. Here's just 3 simple ways that I'll describe it. One is that they pick up DNA from dead cells and they incorporate it into their own genetic repertoire. Number 2, they can have cell-to-cell transfer of genes. And number 3, bacterial viruses, bacteriophage, can inject DNA that they have picked up from a cell that they have already infected. So very clever ways of acquiring new DNA, particularly antibiotic resistance genes. Next slide. So we have also just want to introduce a couple terms that you may hear later in other presentations, but cross-resistance. This means if you're resistant to one antibiotic in a class, that bacterium is resistant to all members of the class. So if you have a macrolide resistant to erythromycin, it's also resistant to all the other members in that class too. Co-resistance includes resistance to different antibiotic classes that are genetically linked. It's like railroad cars on a freight train. They're all linked together. They have tetracycline resistance, macrolide resistance. Those two travel together. They're linked. Use of one antibiotic, a tetracycline, drags along the macrolide. So that's a very important consideration when dealing with resistance. And the last one is multi-drug resistance. You can have 3 or more antibiotic classes, up to 5 or 10 even, and they're all linked together as is in the co-resistance section above. You don't want to have that kind of antibiotic anywhere around because there's very few treatment options in that case. Next slide. So how do you know or assess whether a bug is antibiotic resistant or not, especially with regard to foodborne bacteria such as Salmonella and or Campylobacter? This is done in the laboratory where dilutions are made in test tubes or plates. You add the bacteria and you see what is the lowest concentration which is inhibited by that antibiotic. If there's no growth inhibition, it's going to be termed resistant. And if it's got some inhibition, then it's called susceptible. And those terms, susceptible, resistant, are really directed to the physician, the veterinarian, the clinician, the researcher, because they're all based on the amount of drug that can be administered to a host, whether it's a chicken or a human. And in this case, talking about trying to treat an infection in a susceptible way means you've got enough drug to cover the MIC of the bug. And resistant is just the opposite of that. That your MIC is very high. You cannot get enough drug to be effective, so you will not have a successful treatment outcome. And I put a little note there at the bottom. These are talking about foodborne bacteria so far, but there's no approved breakpoints or MICs for poultry pathogens. Those have not been defined by the groups that do that sort of work. So there is a little caveat there that there's not as much information for making those treatment decisions. Next slide. So let's move into the regulation and risk analysis, and I'll try to pick up the pace a little bit here so we can get this all done. Next slide. So we've seen this one. And so what happens, remember we got the selection, the survival, and then the sickness. And Transform's really working in that first box there in the first circle. So keep that in mind as we work toward reducing foodborne antimicrobial resistant bacteria. Next slide. We could look at this another way and say, well, gee, that's just 3 steps. There's more, Tom. Yes, that's true. And here's a list of them all, but there's still more than that. The idea is that as we go through a risk analysis and a risk assessment, you got to account for all these steps, but the further you get toward the human, the less makes its way through. There are hurdles for transfer, but we don't have a standardized what's the biologically significant percentage for any given individual. But we have to keep these steps in mind as we do the analysis. Next slide. WHO has looked at human antibiotic resistance from human pathogens, and as they put together their documents, they have determined that fluoroquinolone-resistant Salmonella and Campylobacter are 2 of their priority pathogens, foodborne, of course, and fluoroquinolone-resistant. So as we consider what that means going forward, those are of high concern for the human medical community. Next slide. So how do you go about saying, oh, well, gee, what do we do about that sort of thing? This is where we turn to WOA, OIE formerly, and look at their documents about risk analysis processes. Here's a guideline, a roadmap of how to do the risk assessment for antimicrobial agents used in animals. Next slide. Next slide. Oh, you already got it, sorry. Let's go back one if I can. Okay, we had that one, the risk analysis. Let's step ahead. There we go. Okay, so now we're talking on the resistance, and we're down in that lower box, and that will be where we get into the risk analysis. That's what we'll say on that one. Next slide. Okay, so here's the resistance risk analysis outline. So we've talked a little bit about all the pieces here, that selection, the survival, and the consequences of sickness in the 3 steps. Now we'll talk a little bit about risk management and risk communication, which complete the risk analysis paradigm. Next slide. The guidance that regulatory authorities are given to approve the use of antibiotics in food animals has to do with that WHO document, the medically important antibiotics for humans. And so these are requiring veterinary or professional oversight, and the critical important ones need to be minimized in their usage. They're not medically important. They still have to go through the review process of approval, but you have greater latitude to use them because they're not used in humans. It's only when they have the shared classes between humans and animals, and in our case of concern is poultry. Next slide. So here's the document that the important list antibiotics are listed in as defined by expert panels. And the next slide has the OIE list. Similarly, antibiotics are important to the veterinarian and the veterinary community. So there's a list of each that have to be compared and contrasted. Next slide. I'm not going to go through all these. What I want to show you is how these match up. So we have our critically important, highly important, and important, but they're all lumped together as medically important. So you can see that as you go through this, the red, the kind of mauve, the yellow, and this gray, those are all on the human side as medically important. So they have to be studied, the risk analysis done, and appropriate risk management options done. Only in the green are those not used in human medicine. They're not important. So there's a lot of work to be done because of that shared class philosophy. Next slide. And to wrap up a little bit here, there's also an antimicrobial administration that has to be considered. We're all familiar with, disease when it occurs. That take that red line, pathogen causes disease. We try to either prevent it, which would be the green arrow. You see a little blip maybe just there, just a little rise, not much. When it's metaphylaxis at blue, you're treating after the infection is noticed, and you hold the infection in check. And finally, the dark blue indicates that would be therapeutic. You're trying to rescue the animal population from a total disaster here. So the key thing for resistance is you're probably using a very low concentration of drug for prevention and a higher concentration for treatment. The lower concentrations of drug are favorable for selection of antibiotic resistance, whereas the higher concentrations are not as selective for resistance. So that's important when we're considering the foodborne pathogens that are bystanders during the treatment process. Next slide. So we just talked about these three major indications for use, and you'll find those on product labels. Growth promotion is not very well received by the regulatory authorities, pretty much now limited to those that are of not importance to human medicine. Next slide. So this pretty much is just another way of telling you what I've already told you, that anything that's medically important has to be closely looked at, no matter whether it's treatment, control, or prevention. You can use growth promotion only for those antibiotic classes not used in human medicine. Next slide. So what you'll have here is guidance for industry would, go from the most restrictive label, where it's very tightly controlled, need veterinary oversight and maybe single animal use, which is like injection, to the least restrictive, to on-farm use, where it can be used under just basic appropriate use conditions. Groups of animals can be treated here. So the guidance that's on this slide is then reflected in as risk management on the product labels. Next slide. So the product label, as you find it on the bag, the bottle, or whatever, tells you all the information that as a prescriber, user, veterinarian, farmer, to properly administer the antibiotic. And this would fall under that risk communication piece. So now we've completed the full risk analysis section, the risk assessment, risk management, and risk communication. Next slide. So as we come back and circle around to close out here, We talked a little bit about all that happens in the risk assessment, the risk management, which for our purposes here is the labeling and other practices that would be associated with the best practices document of keeping animals healthy in the first place, and risk communication. So again, we're really focusing in that One Health foodborne transmission. Next slide. So we've seen that one. That's where Transform plays. And then we'll wrap up here with the final slide. Next slide. So here's the take homes. You can keep these as like, this is what Tom said. Bacteria can become resistant by acquisition of resistance genes or mutations. We're really looking, bullet 2 here, the foodborne antimicrobial resistant bacterial pathogens. We're not focusing on treatment and the pathogen, disease pathogens of the birds. And then, but when you need those disease interventions, you have to use them appropriately, follow label directions, and that will preserve efficacy, not just against the farm level, but also minimizing selection for potential foodborne resistant pathogens that could enter the food chain. The second key bullet, regulation and risk analysis. The risk analysis process is used to basically guide the regulatory decisions around labeling, and those regulations will help to reduce resistance of foodborne pathogen populations so that the ultimate transfer from poultry into the food chain and ultimately to humans is minimized to the best of our ability. So thank you very much for the opportunity to share a little bit on resistance and the issues and basics. So with that, I will pass it on.
Thank you very much, Doctor, for your intervention. Recordarles que esta iniciativa del Proyecto TRANSFORM está organizada y coordinada por el IPC y en la cual la Asociación Latinoamericana de Avicultura se encuentra trabajando de manera activa para fomentar el uso responsable de antibióticos. La unión entre actores es fundamental en el enfoque de una salud debido a su capacidad para abordar de manera integral y colaborativa los problemas de salud que afectan tanto a las personas como a los animales y el medio ambiente. Este enfoque reconoce que la salud humana, animal y ambiental están interconectadas y deben ser gestionadas de manera conjunta para prevenir y controlar enfermedades, especialmente aquellas que tienen un impacto global, como lo ha sido la pandemia, por ejemplo, de influenza aviar, igual el uso resistente de antimicrobianos. Y para dar cierre a este espacio informativo y educativo, le cedemos la palabra al señor Dennis Erpolding, asesor científico y comercial para la IPC y el proyecto TRANSFORM, con el tema implementación en acción. Dennis.
Thank you, Dania. Appreciate it, and appreciate the deep expertise that Tom has shared with us that we'll reflect on here as we think about the scientific approach. So next slide, please. As Tom noted, we're really looking at the antibiotic resistance, the foodborne one, and the concern we get into globally is looking from a transform standpoint, as we call transboundary, where it causes territorial boundaries, and zoonotic, that transfer between animal and human. And Tom has talked about how we mitigate or lessen that approach and minimize that approach in what we do on the farm level. Next slide. I'm going to just emphasize a couple key points as we think about this thing globally and what IPC is doing, as well as what we're doing through the Transform project. One of the key learnings that we've really learned globally is the importance of definitions. And so I'm just going to repeat and emphasize what Tom did is recognize the word antimicrobial, the word antibiotic, and the word anticoccidial. Antimicrobial is the broadest. Antibiotics very specific. Unique to poultry is the anticoccidials. As we as IPC engage, whether it's through codex or other things, we're very adamant about making sure these definitions stay very clear because that's important to us from an industry, that we have consistent definitions that we use globally. Next slide. Aligned with those definitions, also importantly, is things that Tom noted, we call therapeutic use, and then that includes treatment control and prevention. Treatment would be the curative. Control would also be metaphylaxis. Prevention would be prophylaxis. These IPC definitions are part of our stewardship paper that we have, and holistically. There's actually a definition part in our antimicrobial stewardship paper that goes through all these deals. Importantly, these very much align with the definitions of the World Organization for Animal Health, formerly OIE. And so I think importantly, as we work with stewardship principles, for us, it's important to keep consistency in definitions so we're consistent in what we're doing from a farm practice standpoint. Recognizing as we think about stewardship principles, it can take time and years for us to go from drafting to implementation. So consistency of definition is very important. And we're very pleased that the World Organization for Animal Health, the Codex Task Force on Antimicrobial Resistance, of which IPC has been involved in, has remained consistent in these definitions. So just calling that out. Next slide. Also, as Tom noted, very importantly that we differentiate residues versus resistance. Tom has talked about how that works into the regulatory process. And ultimately then as we think about residues, we're dealing with ADIs, MRLs. We think about resistance, we're thinking about risk analysis. Often people will use these words interchangeably or sometimes confusingly. So I think as we think about definitions, these things are important for consistency and understanding, and why Tom went through the more depth and detail. Next slide. As much as Tom went through the depth and detail on the resistance, I'm just going to highlight one key point as we think about the ADIs, MRLs, which is more dealing with the residue component, is we want to make sure under the ADI, which is acceptable daily intake, all residues, whether it's a marker residue, metabolites, or what might be an unknown part, those are within we call the maximum daily intake, which is under the ADI level. So again, we're not going to get a lot of detail, but just for reference, those things are part of the regulatory process that Tom noted. So importantly, once things go through the regulatory process, whether it's a residue we're dealing with or the pathogen and resistance, those things have been addressed from a public health, food safety standpoint as part of regulatory review process. Next slide. Now let's come back to our stewardship principles that we gave a very much highlight on the last time. I'm going to just go a little bit step deeper as we think about now how this ties back into stewardship principles. So recognizing the science that Tom has talked through, recognizing the stewardship principles, we're going to just dive one step deeper in each of these core principles in what we're doing as think about implementation. And this is what we're trying to do now as we work through the TRANSFORM project, take this from concept to implementation. And we'll just talk through a little bit more in depth on those. Next slide. So think about the first principle. Basically, we take the risk analysis approach. And that really highlights what Tom has just gone through about that whole risk analysis. And through that, we get into what we call this hazard characterization, so that risk profile, the risk assessment, which Tom walked through very nicely, the risk management, that's where regulators decide to the extent we approve and then what controls are approved. And ultimately, the risk communication side, that's where we need to make sure whatever that label has becomes part of that risk communication that goes to users and things of that sort. I think ultimately, as you think about the regulatory process, the drug sponsors actually have to provide the data and information on the risk assessment, and then they work with the regulatory authorities to decide the appropriate risk management conditions that become part of the product label. As we think about the on-farm where the action happens that Nicolas mentioned this time, Ricardo's also mentioned the last call, ultimately we want to make sure that we at the farm are taking the proper actions based upon the label guidance as we work with our veterinarians from the user standpoint. Next slide. So think further about that risk analysis. If you think under it, we also have the important part that IBC will look at understanding when and why we use that antibiotics. So that becomes this veterinary assessment on therapeutic use based on disease progression, diagnostics per label, and timing of administration. The second part is understand and control which antimicrobial is used. So that becomes the antibiotic choice based on importance of ranking, disease diagnostics, efficacy, and things like that. Again, the things that Tom talked through. The third part is understand and control how antimicrobials are used. So importantly, we're following label directions on dosage, maintaining records, handling use, disposition, as well as ultimately monitoring effectiveness. Then the final part is to transparently communicate our actions. So working with, as we think about internal tracking of reporting, reviews, and insurance, so what we do with maintaining consumer confidence and stakeholder confidence. So this ties into that whole first stewardship principle of risk analysis and what we need to do. Next slide. Now we go into the second stewardship principles. That's we talk about management practice around biosecurity, housing, nutrition, hygiene, preventive measures, vaccines, how we enhance animal care. That's really where we have the best practices that we've talked about that Nicola referenced. Next slide, I'll go through those a little bit more. And this, what IPC did in 2019, is after we had the stewardship principles, we recognize the importance of taking this to the next level. What does that mean we do in the farm? So we worked with our IPC experts through the Animal Health and Animal Welfare Committee and looked at stockmanship training, biosecurity, external, internal, bird environment, hygiene, housing, hatcheries, flock health and welfare plans, nutrition, antibiotic medication, the stewardship principles, restriction of growth motion, data collection. So this become our effort to build out from a concept of stewardship principles to how we actually implement that at the farm level. And so as we work through the Transform project, what we're doing now is trying to take these best practices, this guidance, into how we implement that at the farm level. That's a key goal as we advance our work on the antimicrobial work, which is the second stewardship principle that we have. Next slide. Then the third slide is compliance with natural authorization. Tom has walked through this nicely and what that is. That ultimately comes to the label where you have the brand, the product, the caution, the description, the indications, dosage administration, warnings, manufacturer, things like that. So importantly, we want to make sure that we as a poultry industry are compliant with whatever the natural authorizations are. And as Tom noted, there's a stringent regulatory review process in that effort that looks at both the resistance standpoint, but also the residue standpoint. Ultimately, that comes into what's on the label, directions, and guidance. Next slide. Now, if you think about the fourth stewardship principles, we talked about the critical importance for human medicine, the chart that Tom talked about. It only be used for therapeutic purposes under supervised veterinary diagnosis and oversight. We do recognize veterinary infrastructures are different globally. So the goal is ideally a veterinarian, paraprofessional, and things like that. But I think the important thing here, those most medically important for human medicines in classes, whether critically or highly important or important, we want to minimize their use, have their use only be therapeutic use, as Tom mentioned, which is part of the regulatory process, so treatment, control, prevention, ideally under veterinary diagnosis and oversight. So those really try to elaborate a little bit more of our stewardship principles. So as we think about the Transform project, we're taking the IPC stewardship principles and how we're taking those now into more we call the implementation phase, which is what the program that we're into right now. Next slide. This just tries to summarize, you think about the human use medical categorization. Tom walked through that. So as we think about it, ultimately the highest to lowest, you might say, as we think about how we selection on use, and that's whether on humans on the physician side or animals on the veterinarian side. Those that are highest important, you try and minimize their use. And you look at diagnostics, veterinary oversight, and the more important, the more you want to restrict use, or actually, if possible, on the animal side, avoid use. So it just tries to frame this up where we're relying upon that professional advice. For us, it's veterinarians. On the humans, it's physicians. But importantly then is how we work in this process. I will note, as Tom noted, as we think about antimicrobials, which is unique to the poultry sector especially, they're not medically important for human medicine. They're not used in human medicine. So they are one that's probably more discretion, specifically as we think about resistance, where the use in animal does not create an issue. So in some ways, you might say they're one of the most easiest one to select from a use standpoint, since they are for parasite control, which is critical from a coccidiosis control in poultry. But again, our goal is to be a holistic approach on the stewardship principles. And as Annie shared on our last talk, we take that through the Transform, a holistic approach from research, the farm management practices, nutrition health practices, and the stewardship principles. All this ties together in a collective effort. Next slide. And with that, I thank Tom for his scientific introduction, for Ricardo and Maria for our introduction, and Dania. And now I think we will open to discussions and questions. Next slide, please. There we go. Back to you, Dania.
Thank you very much, Dennis. Ahora haremos espacio para preguntas y respuestas. Pueden realizar sus preguntas a través del chat y aprovecho este espacio para nuevamente agradecer a Copantres por su apoyo con la traducción simultánea. Daremos un minuto para recibir preguntas.
Danny, and maybe while we're waiting, I'll just make an observation for Tom as we've got our scientific expert and Tom's traveled globally. He's been involved in regulatory processes. And I know, Tom, we just had like the UN high level meeting coming out of on where they're updating things. As you learn about the impact from the animal to the human and the data that's out there, what are some of the key learnings? Like you talk about what we do on the poultry side, the farm side, and the human side. But as we look at the impact that we have on the animal side, obviously what we do affects a farm. Have we understand better those steps and what's involved and the real impact we're having from the farm level and what we do on resistance and how we minimize that?
Yeah, great question, Dennis. Thank you. There's been many, many learnings over the years here, and it used to be pretty much a straight line. You would look only in the animal and say, aha, I have a resistant bacterium. And then you'd find that same kind of resistant bacterium in a person, straight line, 2 points, you just draw it, it's really easy. Well, what we've come to realize is that there's multiple entry points for bacteria that may appear similar that cause the same kind of disease. So it could be, a water source instead of a meat source, for example. But the finger's always been pointing at animal agriculture. So now with the implementation of One Health, where that whole wiring diagram that I had shared is coming into play, new understandings are emerging that this is even more complicated than 2 points to making a straight line. So I think by now we are getting other disciplines drawn into the whole AMR situation, lending their expertise to try to help sort this out. So it may be more than just booking kind of intervention that somebody would recommend. There's many other things that are being brought forward that also should be done to basically protect public health. So to me, that's been the most interesting thing to develop is the groundswell, the number of people that are engaging through a One Health paradigm.
And if we're still waiting, maybe another comment I would just, Tom, you've obviously worked with regulatory authorities globally. We're recognizing laws and regulations are evolving, but at this stage, you think about how many different regulatory authorities actually have the capability and capacity to do some of the thorough risk assessment. The risk, like what you've talked about, is think about globally, recognizing there's time and resources, but how your observation of how that's evolving as think about regulatory bodies trying to do the right thing to make sure we're managing the risks the best possible at the farm level.
Yeah, that's going to be, it's a global challenge, Dennis. Obviously, the US, Europe, the European Union in its entirety, the developed nations have large capacities to do that. But we also have now organizations like WOA and some of the others that are being able to take that knowledge from those countries and start to apply it in generalized guidelines to assist the regulatory authorities in other countries. Bringing pre-approved products, i.e. those that have been on the market, say in the US or Europe, and trying to get those registered in other countries, there's a data package that may allow smoother approvals and interactions and collaborations. So I think there's some breakthrough in terms of being able to take existing products and maximize their international and global applications. What I am a little apprehensive about is the pipeline of new antibiotics, both for humans as well as for animals. There's not a lot in there. So it is essential that we protect and use wisely those antibiotics that we already have. And I think if there's no other motivation, self-preservation is a very good one here.
And it looks like we may have some questions coming up from the group there.
Sí, tenemos una pregunta en el chat. It says, thanks for your presentation. Which is the sense treatment period for an increased dose of AGP in an enteric case in broilers and in broiler breeders?
This is sensible treatment period. Well, not being a veterinarian.
Dr. Tom?
All I can say is that there really is, we've got to stick with the label directions for the products that are available. And, you know, AGPs, as they may be labeled, to go and try to use a higher dose becomes an off-label use. And so that's the kind of challenge that there's a practical, you want to do something to maintain flock health, but by the same token, if you go off-label, that's not a good practice. So it's a real problem, and one that I do not have an easy answer for you today. It's one of those realities that, you know, maybe through further discussions within the association and other practitioners, there could be a workable solution put forward for those kind of circumstances. But it's a tough challenge.
I think, Tom, if we think about our stewardship principles, I'll jump on that, is basically we need to follow the regulatory authority deals. If there's an antibiotic that might say had a growth motion dose in the past, sometimes companies may go back and say there's a therapeutic dose that could be applied for. So eventually you may have a higher dose, but would require a regulatory approval process. So I think under our stewardship principles, we want to follow the national authorization. And also there's what we call extra label use that can be done via appropriate uses, but that's limited coming back to the national authorizations and regulatory systems. So that actually varies by country. But under our stewardship principles, we say national authorizations, regulatory approvals, and follow what's on the label direction. But if we do see a disease challenge out there, I think that's where companies and others can work together, figure out, do we go back to regulatory authority, go through the regulatory process, the risk analysis, determine is there another dose that could be approved through that regulatory body based upon the national authorities process and approval process there.
Okay, thank you. The other question says, phages, formerly known as bacteriophages, are discussed as alternatives to antibiotics. What is your opinion on this?
I'll jump on that one. I had the opportunity to do some consulting work for a company that was looking to have bacteriophages developed for both human as well as animal health use. And basically, I'll give you my assessment. The company went out of business. So it's going to be a real challenge for commercializing bacteriophage in terms of generating the phage, delivering, maintaining its infectivity, and controlling it once it's out in the environment, so to speak. Because the phage, it's a bacterial virus. So the regulatory authorities will allow that in human clinical disease compassionate use programs in people who have tried everything else, and it does work. But in terms of here's your bottle, vial, or whatever, for food animal treatments, interventions, there's too many biological issues with the bacteriophage approach for it to be commercially successful. Could there be something for companion animals? Maybe, but I'm not sure that the investment would be worth the return on that. So it sounds great. It looks really nice in the laboratory. It's fun to work with, but the commercialization process is going to be very, very expensive. And I'm not sure that there's going to be a market for any such product that's developed. Because once it's out, it's out. Everybody's got access to it. There's no control on it.
Right, there are no more questions. No more questions?
Well, I can just, maybe just add on to that. I don't want to prolong this because I know we're just over the hour. But one thing that I have noticed is many companies have lots of supplements and nutritionals, short chain fatty acids, all sorts of things that are not antibiotic that are being administered feed, water, what have you, to food animals. So do those work? I don't know exactly, but they don't get the label claims as do products registered with, say, the government regulatory authorities. They all say they may increase something like immunity. They may increase microbial performance. They may work, but that's in the eye of the user at this point in time. But there's a lot of that sort of product that's entering the market.
I think, Tom, I would just maybe add as we think about coming towards closure here, I think that's where animal health companies or work with nutrition companies, farm management and things like that. We think about that holistic approach we talked a little bit about the last time of understanding the research options, understanding the microbiome, understanding how we keep that animal healthy, vaccines and other things all come into play. So our goal as we think about this from a stewardship standpoint is what do we do from all the way starting with genetics, nutrition, health management, farm management, and collectively those things done right minimize the need for antibiotics. That's our ultimate goal, is having a healthy animal, minimizing the need for them, but yet if there's a need, making sure they're used properly. And that's where the stewardship principles come into play. Thank you, Tom, for the technical knowledge and expertise today.
Appreciate the opportunity.
Thank you. Thank you, Dr. Tom. Thank you, Dennis. Thank you, Nicolo. We have no more questions. So, dar un formal agradecimiento a nuestros conferencistas del día de hoy, al presidente de la International Poultry Council, don Ricardo Santín, y a la presidente de la Asociación Latinoamericana de Avicultura, la señora María del Rosario Penedo. ALA reitera su sincero agradecimiento al IPC por su invaluable coordinación y participación en el desarrollo de este contenido de nuestro segundo webinar. Les invitamos nuevamente a participar en el tercer y último webinar de esta sesión, que tratará el tema del soporte a la implementación del proyecto TRANSFORM, el día 4 de diciembre a la misma hora, 14 horas Guatemala, Honduras, México y 17 horas Uruguay, Argentina. Muchísimas gracias a todos por participar.